Hello everyone, my name is Margarida and my thesis topic is investigating the presence and foraging behavior of the Harbour porpoise at the Blasket Islands Special Area of Conservation. Ireland is known as an important area for cetaceans with 25 species already cited, and these species are protected by national and international legislation, with two of them, the bottlenose dolphin and the Harbour porpoise, listed under the Annex 2 of the Habitats Directive. This annex requires the designation of special areas of conservation, and therefore through Ireland, SACs have been defined, and here you can see the ones for the bottlenose dolphin in blue and the ones for the Harbour porpoise in orange. And this study will focus on the Blasket Islands SAC, which has the Harbour porpoise as one of the qualifying interests. The Harbour porpoise is one of the most abundant species through Ireland. It is highly vocal and produces a type of vocalizations known as clicks, which are grouped into click trains, and these are produced on average every 12.3 seconds. They are characterized by being narrowband and high frequency, which is quite different from the type of vocalizations emitted by other delphinid species. We can also look to the time between clicks within a click trains, and this allows to distinguish some Harbour porpoise behaviors, and previous studies have used the minimum interclick interval of less than 10 milliseconds as an indicator of foraging. Harbour porpoises are also highly mobile species. However, it has been found that they can spend a lot of their time in certain areas, possibly to reduce the time and energy spent foraging in other locations, and this was hypothesized to be an example of area-restrict search behavior. So to understand the effectiveness of SACs, it is very important to monitor the interest species, and traditionally visual surveys have been used to monitor cetaceans. However, there are some limitations associated with this method, as it is highly dependent on the sea state and weather conditions, and also these species spend a lot of their time underwater, which might increase the difficulty to sight, locate, and follow the individuals. So static acoustic monitoring had become a valuable complement or alternative. This method uses self-contained devices which can be placed underwater in a stationary position and detect the click trains emitted by tube cetaceans for long periods without much maintenance. As all the methods, there are some advantages and disadvantages associated with this method, and I listed some of them here, and this should all be taken into account before implementing a monitoring plan or depending on the research questions. So taking all these aspects into account, this study aims by using static acoustic monitoring to get a better understanding of the presence and foraging behavior of the Harbour porpoise at the Blasket Islands SAC, and has as specific aims determine the temporal patterns for these species using generalized estimation equations, explore the foraging behavior and the influence of environmental variables, and lastly, investigate possible differences in the duration of acoustic encounters considering the occurrence of foraging behavior. So the study area, as I already mentioned, is the Blasket Islands SAC. Two deployment sites were chosen from where the data was collected, and you can see them an approximate location here in this map. And the data was collected during almost 1 year continuously using seapods, which are a type of acoustic data loggers, and they were moored to the seafloor using bottom-mounted acoustic release arrays. And here on the right, you can see a scheme on how they were kept underwater. And the frequency was set to detect tonal clicks in a range from 20 to 160 kilohertz, which includes the vocalization frequency of the Harbour porpoise. So after retrieving the seapods, this data can be extracted using the seapod software, which allows to categorize the clicks based on their characteristics and also depending on their likely origin. So this is how the seapod software looks like, and different settings can be used depending on the type of data that we want to extract and also the research questions. So in this case, we only select the NBHF group as we were only interested in the Harbour porpoise, and we also apply the quality filter to the data. So we extracted 3 types of data. The first one, the detection positive minutes per hour, which was used for the presence analysis, but before this data was converted to a detection positive hour, so to binomial format. Basically, each hour of the study period was classified as presence or absence, depending on the occurrence of DPMs during that hour. And we also extract the click train characteristics as we were interested in the foraging behavior, and by using the minimum interclick interval, this behavior can be identified. So based on this data, we created 2 data sets. The first one, the click train based. So basically we looked at each detection individually and classified it as foraging or not foraging. And we also the second dataset is the foraging positive hour-based dataset. So I have here a scheme on how the filtering process was done. So if you follow the blue arrows, you can see how we obtained this dataset. So basically, to each hour with detections, we classified it as foraging or not foraging, again using the minimum interclick interval as an indicator of foraging. As we were also interested in understanding if there were differences in the time Harbour porpoise would remain in the detection range of the CPOD, depending on whether they were foraging or not, we also extract encounters data, and using a threshold of 10 minutes, which means that two detections that were less than 10 minutes apart were considered to belong to the same encounter. And finally, these encounters were also classified as encounters with foraging or without foraging, again using the minimum interclick interval. So we also assigned some environmental variables to each hour of the study period, namely season, dial, tidal phase, and tidal cycle. And these were divided into categories that you can see here on the right, the categories that were chosen for each one of the environmental variables selected. And for the modeling, we use generalized estimation equations, as I already mentioned, as this method can take autocorrelation into account by grouping the data into blocks. And this type of acoustic data has been found to, in some extent, be correlated, as detections that are closer in time have higher chances of being correlated than detections that are farther apart. So we obtain models for each deployment site separately, so we analyze each site individually, and we obtain a presence and two foraging models for each site using the two foraging datasets that I mentioned before. To validate these models, we use confusion matrix and also a receiver operating characteristic plot, which looks like this, and this also gives us a value known as AUC as an indicator of model performance. So now getting into the results, Harbour porpoise were detected throughout the whole study period. However, a slightly higher occurrence was detected at the Inishtookert, where the Harbour porpoise was detected in 70% of the days monitored in comparison with Wildbank. Looking at foraging, more than 5,000 click trains were detected, and overall 18% of these click trains were classified as foraging. And again, if we look at each location individually, a slightly higher occurrence of foraging behavior was detected at the Inishtookert, represented by the foraging positive hour. Now here we can see the results for the modeling analysis. So on top we have the significant environmental variables that were found for presence, and below we have the two foraging models. So I highlight here the significant differences that were found. And as you can see, depending on whether we look to presence or foraging, or also if we compare with the other deployment site that I will show in the next slide, the environmental variables that were found to be significant change, and also the patterns. So just as an example, day was found to be the period with the highest predicted presence at the Wildbank. However, even though apparently they are more present during this period, the foraging behavior, the day was found to be the period with the least predicted foraging. Now moving on to Inishtookert, a similar analysis was done. So again, I highlight here the significant differences that were found. And as you can see, different environmental variables were found to be significant, and also the trends are different. But I will make an overall view later on during this presentation. Now moving on to the encounter analysis, a similar number of encounters was defined at both sites, and encounters were found to be significantly longer in Inishtookert, and also a higher proportion of encounters with foraging was identified at this site. And if we take a closer look to the time distribution of these encounters, encounters without foraging, which are represented by the white bars, even though they are more frequented, they were found to be significantly shorter in duration in comparison with encounters with foraging, represented by the gray bar, from which also the maximum duration of encounters was found, so for encounters with foraging. Now, to summarize the overall trends found, season and dial were found to be significant predictors of Harbour porpoise presence at both sites, and we were expecting these results, as patterns have been detected in other parts of Ireland and the world, with these two environmental cycles considered two of the most important predictors of Harbour porpoise presence. However, differences across the deployment sites were detected, as Benjamin et al. Demonstrated that variation over small geographic scales may occur. This highlights the importance of conducting small-scale studies. Also, this study was the first acoustic monitoring conducted in the area and just covered one area, so it was not possible to obtain a measure of variability between years, and inter-annual differences has been detected in other SACs through Ireland and also in other areas of Europe, and therefore further acoustic monitoring would be necessary to be sure if the trends found during this study are still the same nowadays, or if they change depending on the year. As for tidal cycle and tidal phase, no significant influence of these cycles was found, and we were not expecting this, as tidal cycle and tidal phase has been found to be significant predictors of harbour porpoise presence in areas through Ireland, and considering the bathymetric characteristics of the Blasket Islands, we were also expecting a significant result, so we hypothesized that this might be related with the deployment location of the seapods, as they were deployed in more sheltered areas to prevent any seapod damage or loss, so probably this might have masked the influence of these environmental cycles. As for foraging, foraging was detected at both sites, and this might indicate that in fact there are pre-resources for this species at the Blasket Islands SAC. And you might be wondering why this is important to know. And first of all, because besides knowing that the harbour porpoises are occurring in the area, it is very relevant to understand how and why they are using the area, so that the best conservation and management strategies can be applied, and that important foraging grounds can also be protected. Again, diel was found to be one of the most significant predictors of harbour porpoise, even though the foraging behaviour did not exactly mirror the presence patterns found. So we hypothesized that possibly this foraging behaviour is related with the diel movements of the prey, as these have been found to be one of the main drivers of harbour porpoise foraging behaviour. We should just take into account that only one seapod was deployed at each site, and this results in a relatively small sampling area, which might have led to an underestimation of foraging behavior and also presence. As again, for tidal cycle and tidal phase, no significant influence of the tidal cycle was found, and as I mentioned, for presence, we were not expecting this. Now, moving on to the encounter analysis, encounters with foraging were found to be significantly longer when compared with encounters without foraging, and this might indicate that, in fact, harbour porpoises remain for long periods in a relatively small area when they are foraging, which might indicate area-restrict search behavior. And these were very interesting results, as this behavior has been acoustically described in bottlenose dolphins and common dolphins. So basically, this type of behavior, even though it has already been described in harbour porpoise, it has been mainly using telemetry. So we can see that possibly we can use this type of data also to analyze this behavior. However, there are some limitations to this analysis, and more research would be needed. So, as I mentioned throughout my discussions, there are some general limitations, especially associated with the type of method and equipment used, and therefore some recommendations were made as to conduct other acoustic surveys, monitor adjacent areas to this SAC, and also conduct surveys on harbour porpoise prey, and also the deployment of more hydrophones and acoustic recorders. And just to conclude my presentation and highlight the main results, static acoustic monitoring was found to be a valuable tool to get a better understanding of the fine scale and foraging behavior of the harbour porpoise at the Blasket Islands SAC. There is evidence that harbour porpoise are occurring during all the year in the area, and also foraging in this SAC. However, variation was found depending on whether on the deployment site and also accordingly to some environmental cycles, and this illustrates the dynamic distribution that these species may display that can vary on relatively small spatial scales, and therefore to create and implement the most effective management and conservation plan, this should be taken into account. So lastly, I would like to thank IMBRC and GMIT for giving me the opportunity to conduct this thesis, and especially my supervisor, Joanne, for all the support and guidance provided during these months. And thank you all for listening, and now I pass to your questions.